And since I'm in person, if you do have any questions, just put your hand up. I'm happy to help it. So it can be sort of as formal as informal as you like. So just a quick overview what I'm going to talk about. So if we go into just basically genetics 101, so I'll be giving a talk on a personalized therapy. So this is actually trying to look at manipulating the DYRK1A gene to try and increase the amount of protein. So I thought I'd just give a nice 101 for those who potentially didn't do genetics. If we think about the DNA in someone's cells as the hardware of a computer, you're born with that. So in the case of DYRK1A syndrome, you're born with a mutation in the DYRK1A gene. A process called transcription happens, which creates a molecule, a single-stranded molecule called RNA. And if you think about this as the software of the computer, this is continuously turned over. It's continuously updated. And it changes depending on the context of the cell or the situation, or even just the context. So if you're cold in here, your body will be reacting to the little things, and some genes will be turning on and some will be turning off. And then a process by which is called translation makes a protein. So the RNA has all the instructions needed. And if you think about it as a Word document, the protein is actually the Word document of the book that you would get out from that software. So what we're going to do is we want to try and manipulate the RNA. There are certain therapies that you can attract, which I'm sure maybe some of you have heard, called gene therapy, which potentially can manipulate the DNA. So that's CRISPR. At the moment, it's probably not a therapeutic tool. There's too many issues with it at the moment, but potentially maybe in the future that could happen. So what we want to do is we want to manipulate the software, because if anything does go wrong with our drug, we can stop it, and therefore the RNA will just go back to the way it was before. So DYRK1A syndrome, obviously the DYRK1A mutation leads to a loss of function of the DYRK1A gene and protein. And this is known as haploinsufficiency. So in genetics, like this is a simplified version. It basically describes a genetic state where a gene is inactivated and the remaining functional copy, so we get one from our mother and one from our father, cannot produce enough protein or gene material to preserve normal cellular function. So this is not the case for all genes. So for the most part, genes will act like, if we think about it as a Tesla, you have a battery that powers the car. If the battery is 50%, the car will still work, right? In the case of haploinsufficient genes, think about it where you have two batteries. One solely powers the front and one solely powers the back. If you have one, and in this instance, the car is, for this analogy, is a four-wheel drive. So you need both permanently. So in this case, and then in the type of a gene like DYRK1A, you only have one battery that's operational, and therefore the other battery is not working, and then you'll be dragging your car along, so in that case, causing lots of dysfunction. So the DYRK1A gene and protein, because obviously I'm going to be talking to you about manipulating the gene and protein, I thought I'd just give you a bit of background about what it does. It's a kinase which basically functions by either activating or inactivating by adding a specific group, like a chemical group, onto another molecule. So by doing that, it either switches it on or it switches it off. These types of proteins are particularly widespread, so that's obviously why in the case of DYRK1A syndrome, we do see such a widespread of phenotype or issues. So obviously we've heard, the previous talk was in the gut, and the previous talk was in the brain, but I know that there's also other issues that occur. And DYRK1A has a huge role. These proteins, these kinases, they generally affect a whole lot of other kinases or other proteins as well. So you can see here, we've heard about cell cycle, cell signaling, which has probably got to do with some of the gut issues, cellular differentiation, which we know is an issue in terms of microcephaly. Apoptosis is about cell death. Axonal transport, which I think also Helen highlighted. synaptic plasticity, and that just talks about the connections between the brain. And when Martin was talking about, hopefully, this is something that we can tap into irrespective of hopefully the child's age, because I'm of the same opinion that I think the brain is a lot more plastic than we think it is in terms of like it can be remodeled. Perfect example is someone in their 80s learning how to play piano. It's a lot more regenerative than I think. But that's at least just mine. This is just about shuttling things in and out of cells, and then splicing and DNA repair. Those are all about how genes are put together. And splicing, I'll touch on that because that's actually a process that we use to manipulate genes. So this is what it is. So genes, if you think about them, they're basically words on a document that provide instructions to make a protein. So what we have is, this is, if you can remember back to the first slide, this is your DNA, it's double-stranded, and this contains all of your genetic information. So what it has is, it has these two regions within it, which one is called an intron and one is called an exon. So if you think about the exons as the words on the document, and the introns are just the spaces between those words. They do have function. It's not needed to know what those functions for the purpose of this talk. But really what happens is there's a process of splicing before it makes that document, where these spaces need to be removed and then precisely joined together. So in the case of DYRK1A syndrome, it's a kinase. I'm not sure if anybody's heard of like that lock and key mechanism for an enzyme. You basically, an enzyme has a specific shape to it, and that shape allows it to bind onto other proteins in a similar how a lock and a key meet together. So in this case, we have the full length transcript here with exons 1, 2, and 3 nicely together, and you have a protein that has a specific shape. In the case of the mutation that I'm talking about today, and the DYRK1A patient that I'm talking about today, they have one of these regions missing. They have an exon missing, which basically removes a bit of genetic information. So if you think about this as a story, they've removed the chapter of the book, you sort of lose the context of the story. In this case, basically, you lose a bit of the information and it will change the shape of the protein. And unfortunately, that then leads the protein to be non-functional. And if we go back here and check the roles that it plays, if you have one copy that's non-functional, you can see that lots of these will be affected. So today I'm talking about a therapy targeted to a single patient, but I do want to highlight, there's going to be sections that will be highlighted in orange, which this will have great implications on the broader community, and hopefully everybody as a whole. So the mutation I'll be talking about is a C1072, and that just, I'm sure everybody knows what those are. I'm sure a geneticist provided that to you. But basically what it is here we have a mutation, one base outside of one of these regions. So that's what the -1 represents. And it's a G2A, and that's a base. I wanted to highlight why that's important. So in terms of determining the exact boundary, or shall I say the start and the finish of this coding region, you need these bases here to tell a system in the cell to split it there. And in the case of this mutation, here you need to recognize basically is if the letter comprises the whole space, that means in humans that should be 100% conserved, i.e. that 100% of people should have that base at that position. If that base is not there, it cannot recognize the start and the end of where it is. And basically you lose that genetic mutation. So this work was done before I joined n-lorem. Initially, we thought the mutation might lead to a different base being selected for the G, but we can see here, so in here you can see that we have an unaffected skin cell, an unaffected neuron, and there should only be one band here. But then here in our patient sample, we got two bands, which represents one of the full copy and then the lower band represents a bit of the genetic information that's missed. And then this was confirmed with sequencing. So this patient has a complete loss of exon 8. So this is what we're going to try and activate. It's a long shot because these sites only work 0.06% of the time. Here you can see the percentage that you should have an A and a G. In this case, we'll have an A and an A, but I'll show you some data later on that's hopefully looking promising. So just represented to you as a gene in a line, and then a protein in a line. So obviously proteins, especially the DYRK1A, they have these specific regions where I believe Helen would have shown you the difference between the frog protein and the human protein, and you saw all those letters very similar. So that's what we like to call conserved. This kinase domain is extremely conserved between all species, and this kinase domain is responsible for its function. So in our patient, we basically lost this chunk of that kinase domain, and the entire kinase domain generally is required for the activity. So how are we going to actually get this back in? So what we're going to do is try and manipulate the gene or trick the cell to, instead of using these code, use one of these. So how do we do that? We use them by these molecules called antisense oligonucleotides. I'll just call them ASOs from now on. So they're short, single-stranded synthetic nucleic acid analogs. So that basically just means they're like a piece of RNA, but they've been synthetically modified so that they can withstand the resistance of your body to degrading it. They're typically 14 to 25 bases in length. I mean, so that's the amount of the genetic code that we need for that. The reason that is so that we can make sure that they don't bind into anywhere else in the genome. So it's like a password. If you had 2 letters on a password, it would be very easy to sort of bind. Same thing, if you just have 2 letters, it will be very easy to bind into different places of the genome. So the 15 to 25 accounts for the fact that you shouldn't really find another 15 or so exact match sequences within the genome. If we do, these are accounted for and then they're removed. They bind by a typical DNA binding to each other, so it's really strong and it doesn't sort of bind to anything else. It might bind proteins, but it shouldn't just free float around and bind to random pieces of genetic information. It should only bind to any perfectly matched. They're designed to be exquisitely selective and specific. So a lot of the drugs that you find will do either increase something or decrease something, but they're not really selective. In this case, because of the way that we design them, they're specifically targeting only that specific region that we want with that gene. And in this instance, it's DYRK1A. So in the case of DYRK1A syndrome, we need these ASOs to be capable of increasing gene and protein expression. Most of the time, ASOs have only been used to decrease protein expression. But what we're going to do is tap into that natural process of splicing. So splicing is a very nuanced function in the cell. It is delicately balanced between positive and negative selection. And in the case of the mutation, it just tipped that balance into the negative selection to remove that genetic information. So what we want to do is we want to switch that selection back to a positive and try and increase the amount of exon selection, and then hopefully increase the amount of protein, the amount of function, and then hopefully overcome the mutation. So in this case, what we'll do is, again, if you remember here, so these are the little introns. We have the exon 8 here represented. The mutation is slightly just in the intron. What we'll do is we'll design an ASO to that region or the surrounding region to tip the balance in favor of what we like to call exon retention or exon inclusion. So you can see here with that, you have a loss of function of the protein. Hopefully what we'll be able to achieve is an increase in this amount of transcript and then increase the protein size and its function. So the relevance to this is this would be the first evidence that a patient with DYRK1A syndrome can be treated with an ASO. So, you know, in theory, these are fantastic tools. They are clinically approved by the FDA for a single patient as well as a broader community. So I'm not sure if you know of Duchenne muscular dystrophy. They're approved for 33% of those patients. Also for spinal muscular atrophy, there's approval. And the foundation where we work, as I said, we're more on the rare, ultra-rare mutations, i.e. a mutation of one. But we've helped them. So these are in patients and they are making a difference. So if we can prove in cells that we can increase the amount of exon selection, and that leads to an increase in protein, then we're well on our way to treating our first patient. So in terms of this, I just wanted to highlight, just to show you a bit of data. I try to keep a lot of the data, scientific data as well in these, because graphs, I don't show the relevance of them sometimes. Here, what you need to do is we have, as I said, you have one copy from your mother, one copy from your father. So here we have a representation of the blue graph is the healthy copy, and the disease is the unhealthy copy. So in this instance, they're represented equally. The whole point of what we're trying to do is decrease the amount of the disease copy and increase the amount of the healthy copy. Now, the idea in terms of DYRK1A is how much do we need to increase this by? I'll touch on that in a second, but what I did want to bring to you here is what we did with these cells as well was we put them through a process called RNA-seq, or whole transcriptome sequencing. I'm sure a lot of people have had a whole exome, so that's where they sequence your DNA. I haven't been able to find any public RNA-seq data that's been compared to controls. So what we did is we compared replicates of we have skin cells from this patient. So what we did was we looked at the entire RNA profile, so the entire gene expression profile of the little patient with DYRK1A and healthy controls. So what we see these are all just the pathways that are dysregulated. So the nice thing is we wanted to know whether we can use skin cells as a conduit to study this disease, because obviously it's more of a neuronal disease and one that affects at least neuronal signaling, it seems, and especially maybe from Helen's, also then the downstream effect of those. But from all those processes that I showed you that DYRK1A is involved, you can see that they start hitting here. So DNA repair, protein transport, chromatin remodeling, cell cycle, RNA processing. So the good thing about this is skin cells are a lot easier to obtain. They're a lot easier to work with, they're a lot cheaper, and they're a lot faster. So we know here that, to my current knowledge, this is the first transcriptome data, and it highlights all the dysregulated genes between the sample that we have and control samples. That seems to match up nicely with the function of DYRK1A. So at least we know that these cells then are a good conduit if we don't have access to iPSCs or these stem cells. So with that, as I said, I showed you here where this graph, we did take these samples and also include them in the RNA-seq, because I wanted to see potentially if we are doing this, are we then having a downstream effect? It's okay to increase the amount of DYRK1A, but hopefully then we will increase all these downstream effects. So this is just what we call an NDS plot. The further away something is the more dissimilar they are. So here we have all the replicates of our untreated sample, patient sample. And in here are clustering with a healthy control. So I tried to age match it as much as we could. And then in the middle here are our treated samples. So then it was done in triplicate. So what this means to the broader community in terms of this data, it highlights that DYRK1A syndrome has a distinct global gene expression difference. compared to patients, compared to healthy controls. And it shows that by using antisense, we are moving this towards our control samples. So in terms of the aims, and I would just like to thank the the Medical and Scientific Advisory Board for inviting me here today, as they've been very generous to providing some funding for this research. So I wanted to present the current aims that we're going to do for this and highlight how it's going to relate back to potentially a broader therapy for the community. So the first, obviously, is I wanted to analyze the transcript and the protein and make sure that that mutation actually leads to a specific consequence. A lot of times, we'll just get a diagnosis of a specific mutation. So that will be your C1072 minus a G21, and a G2A. And someone will just say to you, oh, that's a loss of function mutation. And I think that's a critical piece that we're missing for DYRK1A syndrome, is by just assuming that every single person who has a DYRK1A mutation has a loss of function mutation, I think is very simplistic. We see such a spectrum of phenotype. We see such a spectrum of phenotype in autism as well. So we cannot rule out that in some cases there may be a dominant negative effect. And that what that means is that the mutated protein not only doesn't function the way it's supposed to, but it also then negatively affects the healthy protein. So in the case of a haploinsufficiency, you've just lost gene expression or protein expression. And in dominant negative, it's a bit more complicated. So I think what I hope to do as well, and with this research, is just find out more of the ways in which the genotype, in terms of the genetic mutation, then leads to the dysfunction in the protein or the gene, and how that then relates back to how we see all of the kids and everybody, you know, who's affected by this disease, the large spectrum that is. So just here's a proof that in this instance, I feel the haploinsufficiency of the protein and not of the gene. There might be a bit nuanced for what that means, but that basically means that the amount of gene product that's produced is similar to healthy controls, and we can show that here. So here's our DYRK1A sample, and then these are two unaffected lines. All you need to know is that these differences that you see, although you might see different heights, it's not significant. So it's not actually relative in terms of like clinically. And in one instance, a healthy individual has more DYRK1A expression at this time, and then in this instance, they have less. So what's happening here? Well, obviously, the amount of gene that's being produced is completely different to a typical haploinsufficient. So what I believe is happening is it's what's known as a dead kinase. The protein is being produced, but it just has absolutely no function. So, like I said, I said that before clicking forward. And in terms of how this will relate back to everybody in the room and relate back to all your loved ones, is the more we understand the consequences of the mutation, i.e. the cellular consequences, the more I think we'll be able to understand how we can treat the disease and how different mutations affect different individuals. The second part is the therapeutic avenue. So this is where we'll be conducting a large screen. These are just taken from the grant. So what I wanted to do is show this visually. Now, n-lorem has pioneered the N-of-1 treatment worldwide. Our foundation is leading the way, and this has been done by decades and decades of research by our founder and CEO, Stanley Crook, who's been a pioneer in antisense as well. So what happens is we'll receive the diagnosis. I just wanted to highlight this for you. It's a lot of jargon in here. But basically what happens is we'll get patient material, we'll analyze the mutation, design the drugs to be specific to that person and that mutation only. And in this case, because all mutations have different consequences, the compounds that we'll be creating would only be beneficial to this patient. But the data that we generate is invaluable. So here you can see, so we screen over 500 compounds initially, and to be fair, we probably screen initially 1,000, but those all get reduced in silico. So by the time the algorithm has removed any potential complicating sequences that might cause off-target effects or might not be suitable for the condition, they're removed, and we're generally left with about 500. We'll test them in the cells in a high throughput manner, and hopefully we'll get 50 to 75 hits that are able to manipulate that transcript ratio. From there, we'll do as many of them as we need. We'll err on the side of caution and probably do all of them. You test for any off-target effects, and then you test for any innate immune response. So make sure that these compounds are safe. 20 of them will then be taken forward in mouse tolerability, and then hopefully we're left with 1 to 3 that we can choose before one of them will then go into a patient. So how much gene or protein increase is enough? And this is a hot topic in haploinsufficiency. So for the most part, genes would function like that, like the tester, like I said. If you've got a reduced amount of battery, it should still function and it should pick up the slack. And in most cases it does, because most proteins, there's other proteins that do a similar job, so they pick up the slack. In the context of a haploinsufficient gene, this stark reduction of function happens when you start to lose gene expression. But you can see by stark reduction, you also get a stark increase. So it might not be that we need full 2-fold increase. We might be able to see clinical benefit at 1.5-fold, 1.2-fold. We don't know. So again, this will be the first study and the first way to determine how much or what level of increase of DYRK1A do we need to be therapeutically relevant to the wider population. Just to give you a bit of background, I come from a lab where before I joined n-lorem, our lab was part of developing the 3 drugs for the Duchenne muscular dystrophy boys. They went from no detectable dystrophin compared to a healthy control to only 1.2% dystrophin. But that gave them the ability to remain walking for almost a decade now for one of the boys, longer than what he should. So I'm not saying that we only need a 1% increase here. That's not what I'm saying. I'm just saying that while the goal is 2-fold, clinical benefit can be seen at other levels potentially. So why is it benefit at n-lorem? So at n-lorem, we have a proven track record of success. Under the guidance of Stanley Crook, he's been a pioneer in the space for decades. He came from a larger pharmaceutical company and wanted to start a foundation tackling ultra-rare disease. We have rapid development of a viable therapy, which can be done from when we receive the cells to when we start to file for IND. So that's when you actually file for registration of that drug to hopefully go into that patient. Can be done between 12 and 24 months, where typical pharmacological development, stay for decades. It'll provide the first human trial of a DYRK1A patient, allowing us to gather the invaluable data into the benefits of this potential therapy, and will provide the first postnatal treatment of anybody with DYRK1A. And then, as we've heard from the previous talk, see if the plasticity, see if we get gut plasticity, see if we get neuronal plasticity. I'm always a half-glass full person, but in terms of the brain plasticity, I think that's somewhere where there might be benefit. And there are other mechanisms by which antisense can work. So these these tools can be used to either increase gene expression, decrease gene expression, change the way the gene is put together. And a technique actually that was first published by Stanley Crook is what's called translation upregulation. So if you remember back to that Genetics 101, for a lot of the kids, we might actually have a true haploinsufficiency phenotype where you have a single base change that leads to a loss of function mutation. And this approach is completely mutation agnostic if you are a true haploinsufficient. This binds onto negative regulatory elements in basically a region that controls how fast your protein is translated. So this has been published. This is not what I'm currently working on, but this is the broad applicability that can come from understanding how antisense affects one patient, then to understand how antisense can affect a whole group of patients. So upon successful completion of the grant, we hope that we'll be able to show upregulation in our patient cells, be able to find ASOs that are very potent, very selective for DYRK1A, and non-toxic. And this will potentially de-risk a mutation-agnostic approach. So in terms of the mutation, I thought I'd just end with an analogy. Translation is like Goldilocks and a 3 start site. So like I said, you have a gene message, but there has to be a start of the gene. There's a compound that recognizes the start of the gene, but a lot of the times there's multiple of these, right? So in the context of Goldilocks, she comes into the room and she's got 3 bowls. The first bowl she'll try will be too cold. The second bowl she'll try is too hot, and then the third one will be delicious. But if you think about that process took a little bit of time to get to, right? So in the case, this is the same thing that's happening in biology. A compound called the ribosome comes and it looks at the start site, but it has to check which is the actual right one. So in the case of antisense, they bind onto these start sites and actually just cover those. And then it leads the system to only then moving to the right bowl. So then what will happen is by removing the options, you basically shunt the system to increase the amount of protein expression. So what I hope to show you today was just that there's a therapeutic pipeline. Although this is an N-of-1 therapy, I hope I've shown you how this can drastically change the landscape of potential DYRK1A therapeutics by providing evidence of a potential benefit. We aim to increase it through the splice modulating AOs, which are already in the clinic. This research will lay the foundation into a broader therapeutic, hopefully, and provide the first evidence that DYRK1A can be treated in a disease-altering manner. Obviously, I'm here presenting on behalf of our lab, but it takes, obviously, a village to raise a child. So thank you to everyone here. And I'd just like to thank the DYRK1A Syndrome Association for your kind and generous donation to this research, and hopefully we'll have some good stories for you in the next meetup in Texas next year. Hopefully this is one of those success stories that we can move forward with. Thank you. I can just show. That's all right.
I guess I had a few questions. So you've been talking about one.
Hello.
You've been talking about one. Is that when you said it's success stories? Is that in trying patients or?
Yeah, so this is an enrolled program at n-lorem. So the goal is at n-lorem is to create therapies for nano-rare, ultra-rare mutations, so N of 1 or N of a few. and provide that drug free for life to that patient. In the context of this, yes. So this patient was submitted to n-lorem and was accepted, and then will go through the same pipeline that most cases will go through. So they'll come into a discovery phase where we'll test the compounds, make sure that they can do what we want, make sure that they're safe, make sure that they don't cause any off-target effects. Then they'll go into small rodent studies to do the same thing, not necessarily to check the activity of the drug, just to make sure it's safe. Because obviously we are going to inject this intrathecally, so it would be through the spine into the brain. We want to make sure that these are safe. And then the regulatory process to which an approval of an N of 1 therapy goes through compared to a commercial huge therapy, say for something like diabetes, is very, very different. And in the benefit of this side is it's rapid. So normally you need to go through phase 1, 2, 3 clinical trials. In this case, because it's an N of 1, dosing the patient becomes the phase 3 itself, while also serving as the therapy itself, if that makes sense, without the need for your placebo-controlled trial, whereby half the patients don't actually receive the active compound.
It's a one-time injection?
No, it would be ongoing. So, like I said in the first slide, your RNA is continuously turned over. Your cell never stops transcribing until it undergoes cell death, and even then it does. So we anticipate that you would probably need to dose quarterly. From other intrathecal programs, it's quarterly initially, but it depends obviously on the disease course. So those things would be probably more spoken to by our regulatory team. I'm more on the research side, where we try and find the compounds. But from what I understand is generally they'll need to be dosed, if we're just speaking frank, for the rest of their life. If you're going to use this therapy. There are other things such as gene therapy. I don't know where they are in terms of DYRK1A. I haven't seen any published research that's looking at a gene therapy for it. But there are gene therapies for other diseases that have been approved, so that is a potential. The one issue, caveat I would say with DYRK1A and gene therapy is because it is a dose-sensitive gene, and obviously it's overexpressed in Down syndrome, and it's thought to cause a lot of the intellectual disability seen in Down syndrome. So if you look at some of the Down syndrome research, they've tried to reduce this gene and seen like a benefit in behavioral issues. With gene therapy, you can't necessarily control the amount of expression that you get because it's delivered through a vector. So in my opinion, what the closest is to a viable therapy would be antisense, although gene therapy is a promising sort of next step.
Thank you.
Thank you.
Any other questions?
I got a question. So
Yeah.
the
RNA?
the RNA, you say it keeps turning over, keeps
Yeah.
developing. So in the DYRK1A children, their hardware is not set up to break that RNA properly.
So basically what's happened is so they have a mutation here in the DNA.
Okay.
That unfortunately is always copied. So this is a mirror image, it's just single strand. So this is RNA is a copy of that. So in the context of the hardware and the software, like the analogy is not exactly scientifically sound.
Yeah.
But I find it's good just for explaining the gene expression. You have a mutation in your DNA that will always get carried over into your RNA, and then from that, that'll always get carried over into your protein.
And that's how you do injection.
Yeah. So if say something like CRISPR was a viable option now, that comes in and that actually changes your DNA. But the issue with that and why it's not a viable treatment option, it's very good for studying mutations. As Martin said, they introduce a mutation by CRISPR, and it's very good in cell models. The issue is it's irreversible. So if you mutate the DNA in the wrong place, every cell that's replicated from that cell will have that same mistake. So the consequences are very high if you get it wrong. While the consequences here are still high if you don't develop the drug properly, which is not something that we do. We take great care in developing the safest compound that we can. But if there are any issues here and you stop this therapy, it stops manipulating the RNA and you can go back. But with CRISPR, if you say dose, whatever cell has been changed, any cell from then on, or daughter cell of that, will be the same.
You do that stem cell research I was talking about.
Yeah, so because we're a non-for-profit, so we obviously don't have the backing of a huge pharmaceutical company and publicly traded company. Neurons are very expensive to work with. So while we do that for specific cases, if we can get away with using skin cells, it's a much faster, cheaper, and more efficient way to screen drugs than to screen them in neurons. But yes, depending on the case, some genes are only expressed in specific tissues. So some are only expressed in the brain. So in cases where we're only looking at a mutation that is expressed in the gene only in the brain, then we have to use iPSCs. But that program will take longer because you need to generate the stem cells. Then from there, you need to differentiate, which can take up to 21 days if everything goes all well, sometimes as long as 6 months. In this case, we get skin cells that grow fantastically. They're rapid. A puncture biopsy is a lot less invasive than some other factors. So we also find skin cells much better.
Great, thanks so much for that presentation. Really appreciate it. I mean, you laid it out just very eloquently and applaud your work. Yeah, I think one thing that you mentioned, really resonated too, and I was really trying to characterize the mutations just functionally. Like, what do the mutations do? Because I think that's a huge gap in where we all are, because we hear, okay, you have this mutation, your child has this mutation, it's either pathogenic, likely pathogenic, as the geneticists would say, and then from where do you go? Because like you said, a lot of what you hear is it's just loss of function, but what we see is that's not always the case. So as like a community, like how can we learn more about our mutations, like just from a functional standpoint? Like, are there labs that work on like just functional analysis, or how can we better understand sort of where each of our mutations are and what they actually do?
Yeah, look, I definitely think I think there needs to be a better connect between, although I work in translational medicine, but there's a very good connect between people who make drugs and clinicians, because generally there's quite a good link, right? But there's not such a good link unless you get a clinician, also a doctor who's a researcher as well, between clinicians and basic biologists. So basic biologists generally will study the biology of a cell or an organ system or anything like that. So I think, yes, there's definitely people working on DYRK1A. In terms of DYRK1A, it just so happens that obviously it's on chromosome 21 and triplicated in Down syndrome and quite implicated in the phenotype of Down syndrome. And due to the broader population of them, I think it's been characterized in a better manner in an overexpression system. So DYRK1A is really nicely characterized in DYRK1A, in Down syndrome, but in DYRK1A, it's not. So I think what we need to do as researchers and basic biologists is get together get as many samples as you can, do whole transcriptome, and actually look at the gene and how it's put together. Because what I found in working in an academic lab, not now with the foundation, was we were a lab on rare disease. We had the same thing. We would get all these mutations where someone's like, oh, you have this mutation. And if you take that for granted, then yeah, that's what you have. But often we would interrogate it deeper, being like, okay, well, actually, like, this is saying you have a single base change. But what does that mean in the RNA? Because how is the RNA put together? How is the RNA then affected? How is the stability affected? All those things affect the amount of gene product. So I think, yeah, what needs to happen is there needs to be a connect between what's out there phenotypically, which I think a lot of people know, but then getting samples, getting biological samples from patients and being able to look at that, see actually on what we call like a qPCR, whether their expression is decreased, or is it like, in this instance, similar, but obviously something else is going on. Because, you know, this specific individual is quite severe. So how is it that that is more severe than potentially a protein truncating mutation, which is basically a stop nonsense mutation, which causes a stop. So I really think, coming back to that question, sorry, that was a long-winded manner, is just, I think anybody working or interested in the space, getting as many samples and then doing a genotype, but not just sequencing the DNA, like actually interrogating it at a molecular genetic level. So PCR, qPCR, RNA sequencing, and other assays, I think, kinase assays.
And then this will just be my final question. So I thought it was going to be interesting when you showed the gene expression, just in comparison with like the unaffected individuals, like, you know, one was up, one was low. Like, was that something you were expecting, or like, I mean, does that give any sort of rise to like why there's just different levels?
I think so, yeah, I think so. So coming back to a disease that I guess is better characterized or more published, so with Duchenne dystrophin, the DMD gene, fluctuates up to 4-fold in healthy individuals, and in DMD it fluctuated, you know, on a spectrum from 100 all the way down. So you get Becker if you had a milder version. From healthy samples in my old position, the sample that we had lower expression. So that's why I initially thought it had DYRK1A, but then we tested it on the ones that we have here, and they were all over the place. So yeah, I think the also caveat that I think, and this is just purely anecdotal, so this is just a thought that I have, is DYRK1A plays a large role in cell cycle, and I also think it depends on when you get the cells as well. I'd like to see if I can do a few cell cycle experiments where what you can do is you can link all the cells up to a specific, you know, phase within the cell cycle, and then you can see how quickly they either grow or don't grow or senesce. Because I've got a feeling, and that's just anecdotally working with these cells, they're small, but they grow. So I'd like to know. It's funny because they think that DYRK1A inhibits a bit of cell growth, but I wonder in a fibroblast, is that the case? So it's also looking at different cell types. But yeah, I think every time we think we know something about a gene, I think something comes up. So while it's well characterized in Down syndrome, I think, yeah, trying to find the level of just baseline DYRK1A throughout the population would be something of great value as well. Just sequence a whole bunch of people like they did with, including on different genetic backgrounds, and see if that does play a role as well. Because obviously in the mouse model, they've shown that the genetic background plays a huge role, obviously. So it would be interesting to see. If you had two patients with the same mutation from potentially two ethnic groups or two sexes,
I have just one curiosity question. This patient, again, like that is more severe case and is a different type than nonsense. What type of mutation is that?
So a nonsense mutation is a base change that basically leads to, okay, let me just go back to this. Okay. So this is the whole gene, right? If you have a specific mutation within an exon that leads to a base change that changes the amino acid in the protein to what we call a stop codon. A protein is read by a start codon, and then all those little amino acids in between until it reaches a predefined stop codon. And then it stops translating, and then the protein is read. And then it goes out and functions. In the case of a nonsense mutation, instead of the stop being at the end of the sentence, if you think about it as a sentence, stop is in the middle of the sentence. So you lose the end of the message. So in the most part, what would happen is most people's mutations are within this kinase domain. If it's a nonsense mutation, and say the nonsense mutation is here, protein will stop there, and the rest of this won't be produced. So basically, you lose this entire region of the protein, or wherever the nonsense mutation is, you lose everything after that nonsense. And that typically, unless, without going into other, typically that is a true sort of haploinsufficiency, because what happens is the mutation itself causes, the cell is very clever, it knows when there's mistakes. It has an internal recognition system to say degrade that gene message, it is wrong. So that's called nonsense mediated decay. So what happens is it quickly will degrade it so that it doesn't produce that wrong protein. So in that, then that's typically a true haploinsufficiency. In this case, now we're getting into a bit more genetics. This exon is in frame. So it means that it can be removed and then the protein can be put together without changing the sequence. So it's a very, very unique mutation. So that's why you don't see a reduction in the transcript, because it hasn't changed the downstream message. It's basically like you've just did a little bit of whiteout and then you stuck two pieces together. In this case, then, that's why you don't see the decrease in gene expression, because that mechanism by which the cell uses to remove that transcript is not activated in this case, because this part is not lost.
Could you explain to me what it means to have a missense variant?
Yeah, okay. So a missense variant then is a single base change in a coding sequence in an exon. So if we think of 3 exons as 3 words, the cat, like I say, 4 words. Okay, so the genes are read in letters of 3, and that makes 1 amino acid. So think of the cat ate, or the cat sat on the mat, right? A missense would be the cap sat on the mat. So it changes 1 amino acid letter. But what it does in the context of a kinase is it'll do something similar to that. Each amino acid has specific biochemical properties which make it fit around the protein. So proteins have this 3D structure, and they're generally coiled on themselves, and they're very complicated. And even 1 amino acid can just kink that folding. And in the context of a kinase or a transcription factor, which affects other things, it affects then the way that they would meet and dock onto the protein that they then affect. So then it's kind of like imperfect docking. And then the imperfect docking then leads to, obviously, it not doing its function in the cell because it can't bind onto its specific target and what we call phosphorylate it. So put a phosphate. So a missense, depending on where it is in the gene, most, in terms of DYRK1A, most missense are in the kinase domain because that is ultra conserved. That kinase domain, like a lock and key, if you damage the key to a certain extent or you change the shape of the key, it would no longer fit in that. So a missense in that sense will change the, typically it does. Sometimes it changes the activity, depending on where the missense is. It might just lose activity because that amino acid is critical. So in the case of DYRK1A, it phosphorylates on tyrosine 321, I believe. So if that is a missense, then it can't activate itself. That's fine.
Is that also HACO?
That's where we don't know. That's where I feel like there needs to be more understanding, because a lot of cases you can have a missense mutation and then you get what they call a toxic gain of function. And that gives it a dominant negative effect where the protein then gains this toxic function. Sometimes it also loses its function to do what it does, but it also then has a negative function. And that can cause other problems. So I think those types of mutations and the nuance of those types of mutations are exactly what we need to look at to see whether it retains its kinase activity or not. A lot of the times as well in research, we use like methods by which you overexpress proteins. I'm a proponent of, I really do also like to think we need to look at the patient material as well. So it's really important whenever possible for researchers to use patient material. It might. Often, I don't know because obviously I'm not too sure when you get up, and I haven't lived that long in America, so I don't know the reports that you get here. But typically, maybe when you spoke to a genetic counselor, they might have provided that.
Sometimes.
Yeah, I was gonna say ours already didn't.
Okay.
Yeah, mine did say so. But what I understood from other families, the report that they got from the doctors is all completely different. Some got, some not.
And sometimes the consequence of that mutation is not known. Like for instance, in this case, for a while we didn't actually know that this mutation led to exon 8 skipping. It was a splice site mutation that could go either way. It could enact another change in the protein that was slightly different. That would have led to what is called a nonsense mutation. But through further investigation, we found that it wasn't the case. So sometimes it's not as clear cut. And that's what I mean by when Oliver asked, how do we relate the genetic diagnosis to how your loved ones present in terms of the spectrum of DYRK1A. I think understanding the consequence of that mutation at the genetic level, not just saying, oh, this is the mutation, or what's cause and effect, right? That's the what's the effect of that on the gene.